Learning goal today is to describe some methods for investigating the abiotic and biotic interactions in ecosystems. Now, quick vocab here. Abiotic factors in the environment is referring to things that are non-living in the ecosystem. For example, you can have soil, you could have gases in the atmosphere, you could have sunlight, temperature, rocks, water. Anything that's not alive in an ecosystem can be called an abiotic factor. Now, biotic factor is just the opposite. These are all the living things in an environment that interact with the abiotic factors. For example, animals, plants, bacteria, anything alive is considered biotic. And now the study of interactions between abiotic and biotic factors in an ecosystem is what we call ecology. So today, of course, we've got our Cornell notes. We should be experts at this by now. We're going to write our notes in this section here. You're going to fill in the blanks, and then in the left side margin you're going to put your questions. This one's a little bit longer than usual. It's going to be 3 pages, and you'll have your summary down here. This 4th page here, don't worry about that one. We'll do that together at a later date. So really today's notes is all about collecting data from the environment. So just how do we know what's going on in any ecosystem? It's so complex. What we have to do is measure various biotic and abiotic factors. And then we look for interactions between those sets of data. So first we'll take a look at measuring abiotic factors in an ecosystem, and generally we divide ecosystems into 3 broad categories. We have the marine or saltwater category, the freshwater category, and the terrestrial or land category. And so here we've got some pictures to represent those categories. We have a marine here, we have a freshwater here, and we have a terrestrial category here. Now it's important to note that each of these categories has its own set of abiotic factors that we need to look at specifically for that category. We'll use the book to find information on these specific factors in a little bit. We're going to do that in groups. But for today, we're going to take a look at some of the main ones. So the study of ecosystems, or ecology as we call it, really is about the interactions of the abiotic factors with the biotic factors. So we really must know about the abiotic factors before we start looking at the biotic factors. And that's what we're going to look at first. So I want you to think about this as we go through these notes. Why do we begin ecosystem investigations by looking at the abiotic factors first? So once we know the abiotic conditions of an environment, for example, once we know about the soil, air, or water quality, only then can we start to ask questions about the organisms that live in a particular area. So this is when we can get into measuring the biotic factors. So some questions we could ask when we're measuring biotic factors are: which species are present? We could look at the population size of a particular organism that lives in an ecosystem. We could ask how productive is a given area, which really means how efficient is a certain ecosystem at gaining biomass or organic material. And we could ask, what is the diversity in a given area? And when we're talking about biotic diversity or species diversity, what we're asking is how many different species are represented in a given community. So think about this. Could you suggest a research question you might pose about biotic factors in an ecosystem? And remember, similarly to how we did this in chemistry class, we can set these questions up in the format, what is the effect of blank on blank? And typically the first blank would be an abiotic factor, and the second blank would be your biotic factor. For example, I could say something such as, what is the effect of air quality on bird species diversity? So there are many methods for measuring biotic factors. The method you choose depends on what you're studying and what type of question you're asking. And just to look at some examples of biotic factors that could be measured in an ecosystem, you may want to do something such as measure plant biomass, measure productivity, which is the rate at which organisms create biomass, estimate population or community size, identify the different organisms in a population, identify what type of organisms are living in an ecosystem, and measure the diversity of an ecosystem. And like I said, we're going to take a look at some more specific strategies in a bit. We're going to do that in groups. So think about it. Hopefully by this point we would be able to list some biotic factors that you might want to measure in an ecosystem. Anytime we're trying to measure biotic factors in an environment, it's impossible to measure everything in a particular area. So what we do is we take samples and then we apply some type of statistical analysis to our data, and we make estimations about the entire ecosystem. And we want to make sure that anytime we're trying to measure a biotic factor, that we select the area randomly to avoid any bias. And what this does is produce a scenario in which all organisms have an equal chance of being captured and accounted for. One biotic sampling technique that can be utilized is called quadrat sampling. A quadrat is simply a square frame used to identify the area that you want to draw a sample from. Basically, you lay this frame down in a specific area of the community that you want to take your sample. Now, the size of your quadrat varies depending on what you want to sample. For example, you might have a 10 by 10 centimeter quadrat size if you want to measure small organisms such as algae or lichens. And you might have a larger, maybe 1 by 1 meter quadrat size if you want to measure larger plants. And then you could also go up to a 5 by 5 meter quadrat size to measure mature trees. So real quick, we'll go over the steps for correct quadrat sampling. First step is you want to map out your study area, make a numbered grid. Then you want to use a random number generator to identify which squares in your quadrat you're going to sample. Then you're going to count the number of organisms in each randomly chosen sample area, find the mean of the number of organisms per sample, multiply the mean by the number of squares in your grid, and thus we can efficiently estimate population size by measuring only a small sample of the area where the organisms live. Now, there are some caveats to quadrat sampling, or some limitations. For example, this only works for non-motile organisms, which means they don't move around, or very slow-moving organisms. There's another issue you would have to deal with if you had two distinctly different vegetation types, or you just had obvious differences within an area that you wanted to sample. For example, maybe you had an area of study that included a forest and then the edge of the forest, and then you had a prairie. What you would do is called stratified sampling. Now, when you have these types of complex situations, the best way to attack it is to really make two separate grids and then deal with each one individually. It's important to strike a balance between accuracy and efficiency. You don't want to spend too much time on it, but you want your data to be accurate. So we should be able to explain how to use quadrat sampling to estimate population size efficiently and accurately. Now let's move on and talk about something called a transect. Now, a transect is simply a line or path usually made with string or rope, which you lay down, and then the organisms are counted that come into contact with that line or rope. Now, the length of your transect varies depending on the area that you're studying. And this is similar to a quadrat. There's no set size. It depends on what you're sampling and what area you're in. So real quick, we'll go over the steps. You're going to lay out your transect. You lay down the rope or the string. Now, there are two types of transects. You have a line transect, which is simply a straight line, and then the organisms touching it are counted. Or you have a belt transect, which is two parallel ropes or strings, generally about 1/2 to 1 meter apart, and the organisms between the ropes are counted. And once your transect is laid out, the next step is to simply count the organisms. Now, there's two methods for this. There's continuous, where you simply count everything along the line. Then you have the interrupted method, where you place quadrats at set intervals along the line. Again, there are some limitations. For example, it only works for non-motile organisms, so non-moving things. I've done transects where we measured plants. These are best for studying changes along a gradient, and you may need multiple transects, usually about 3 to 5, to obtain sufficient data. At this point, we should be able to explain how we use transects to estimate population size. Let's talk about measuring the abundance of non-motile species in an ecosystem. And remember, when we say non-motile, usually we're talking about plants or trees. So 3 ways we can measure abundance of plants or trees is by looking at their density, how close they are to each other, the frequency, which is how many of them there are, or the percent cover, which is how much of a given area is covered by a type of plant or tree. Now, the method that you choose depends on what you're researching. Now, the way that we could measure density is we take the number of plants and we divide by square meters of the area that you're sampling. How we would measure frequency is we would take the percent of total quadrats that the species was found in, and we could measure percent cover by figuring out how much of the ecosystem was covered by a specific plant. This can be useful with things like mosses or lichens, where it can be difficult to figure out where one individual organism ends and a new one begins. So we should be able to list the 3 ways that we can measure the abundance of non-moving species in an ecosystem. Now let's take a look at some ways we can measure the diversity of an ecosystem. There are 3 ways we can think about diversity. We can look at the species diversity, which is the number of different species that are represented in a given community. We can look at the genetic diversity, which is where we figure out the frequency of alleles or genes in an ecosystem. And then we can look at the habitat diversity, which is the number of different habitats that are found in a particular region. And you can see we've got a star here by species diversity. That's the one we're going to take a closer look at now. One method for measuring species diversity is something that we call the Simpson's Reciprocal Index, or sometimes called the Simpson's Diversity Index. And what ecologists try to do is assign a number to a given ecosystem that indicates how diverse that ecosystem is. Now with Simpson's, the lowest value that you can have is 1, and this indicates only one species present. The higher the number is, the more diverse an ecosystem is. And here we have our equation. The capital D stands for the Simpson's diversity number. Capital N represents the total number of organisms of all the species found. Lowercase n represents the number of individuals of a particular species. And this symbol here represents the sum of. So this is the example from the book. Let's just take a quick look at it. We've got 2 ecosystems here. You can see in ecosystem 1, there are 3 different species, and the number of those species are fairly evenly distributed. In ecosystem 2, we have the same 3 different species, but the distribution is significantly skewed towards species A. To figure the diversity index of ecosystem 1, you add up the entire number of all the species, that's capital N, you multiply that by n-1. In the denominator, we have the sum of all the numbers of the individuals of each species, multiplied by n-1. And you can see that we get a Simpson's diversity index of 3.07. The higher this number is, the more stable the ecosystem tends to be. If we took a look at ecosystem 2, we have a less even distribution. Therefore, the diversity index is lower, and the number that we'd get from the equation for ecosystem 2 would be 1.22. Generally, we'd see lower numbers for the diversity index in places that have been polluted or highly disturbed, such as logged forests. Simpson's diversity index accounts for species richness, which is how many species are present, and also species evenness, which is how evenly distributed the population sizes are. And once again, this is generally only used for non-motile populations of things such as plants or trees. So at this point, we should be able to outline the use of Simpson's index. So what about moving creatures? How do we account for those individuals? Well, if it moves slow enough, they can be counted one by one. But things that are more mobile, we use the CMRR method. This stands for capture, mark, release, and recapture. And the steps for this method are as follows. You want to establish your study area, capture a sample of the organism, mark the organism, hopefully in a non-harmful and non-obvious way, release the organism, and then allow enough time for them to reintegrate back into the population. Once they do this, you can take a second sample and count how many of those are marked. Now, I worked on a study where we estimated the size of bird populations. What we would do is mark out a given area, set up nets, capture the birds, put a small metal band on their leg, release them back to the environment, and then we'd come back to the same area year after year. The thing about birds is they migrate every year, but they always come back to the same area in the summer. So we'd put up the same nets, and a lot of times we would catch birds that had the bands on their legs from the previous years. Then we could crunch the numbers with some statistical analysis and estimate the size of the population of different species of birds. So we should be able to describe the CMRR method for estimating motile populations of organisms. Now let's talk about the Lincoln Index formula. There are some assumptions that you have to make for this formula to be accurate. For example, marked individuals can move back into the population unnoticed. The marks will not disappear, and there's no change in population size between when you take your samples. It's equally easy to catch all individuals during both samples, and at least 10% of the marked sample should be recaptured for accuracy. Here we have the Lincoln Index formula. Now with the Lincoln Index formula, you can estimate capital N, which is the total population size. And how you do this is you multiply the number of your first sample times the number of your second sample divided by how many marked individuals were captured in your second sample. So we should be able to state the outcome of the Lincoln Index formula. That's it for the notes this time. Don't forget to write your 3 questions in the margin and complete your summary at the end.